Everyone, today I'm going to discuss about a very important disease of poultry, that is the Ranikhet disease. It is prepared by me, that is Durbojyoti Kumar, a student of 4th professional year, Lakhimpur College of Veterinary Science, and it is prepared under the guidance of Dr. Sampurnanand Yadav sir, STEM professor, Department of Veterinary Medicine, Lakhimpur College of Veterinary Science. Okay, I will start by introducing you about this disease. This disease is a very highly contagious acute viral disease of birds, and it is characterized by respiratory signs and nervous signs, which include wing paralysis, incoordination, torticollis, swelling of head, and socky white diarrhea. This disease was first discovered in 1926 in Java, Indonesia, and in Newcastle, England. So from this place, Newcastle, it got its name, Newcastle disease. This disease was discovered in 1927 in a place called Ranikhet in India. Hence, in India, it is also known as Ranikhet disease. So etiology, this disease is caused by a single-stranded RNA virus called as avian paramyxovirus type 1. It belongs to the genus Ebolavirus and subfamily Paramyxoviridae and family Paramyxoviridae. So there are many strains of this virus, out of which the pathogenic strains include velogenic strain, HERTS33, Texas DB, mesogenic strains K, R2B, lentogenic strains include F, Lasota, Ulster 2C, and avirulent strains include V4. So this is the diagrammatic representation of the virus, which includes 6 proteins like M for matrix protein, HN for hemagglutinin neuraminidase, F for fusion protein, N for nucleocapsid, P for phosphoprotein, and L for large polymerase protein. Okay, transmission. This disease can transmit from many routes, which include aerosol, then oral and fecal droppings of the birds, then from infected birds to non-infected birds, from wild infected birds to domestic birds, and vice versa, and from vehicles or equipments contaminated with the infected droppings from the infected birds, and so on. So this disease can spread very rapidly and at a very fast rate. Pathogenesis. The initial step of this virus is the attachment of the virus with the cell receptors, which is mediated by the HN glycoprotein. Then it fuses with the cell membrane with the help of the fusion F glycoprotein. Then the nucleocapsid enters the cell. In the cell, it replicates in the cell cytoplasm. Then the F protein is synthesized as a non-functional precursor FO, which requires cleavage to F1 and F2 by host proteases for the production of infective viral particles. In some strains of the Ranikhet virus, it may require a post-translational cleavage, which play a major role in the pathogenicity of the virus. So this is the life cycle of the virus, as I have discussed. It fuses with the cell membrane, then replicates inside, and then exits the cell and transmits the disease. So 5 clinical forms of this disease are recognized, which include viscerotropic, phylogenic, or the Doyle's form, neurogenic strain causes the neurotropic form, mesogenic form, lentogenic form, asymptomatically enteric form. So talking about the viscerotropic form, it is the most pathogenic form caused by the pathogenic velogenic strains. And the typical symptoms include depression, prostration, diarrhea, edema of the head, and nervous signs. And mortality reaches up to 100%. Hemorrhagic lesions are seen in the digestive tract. Then the infection caused by this disease is also called as the Asiatic or exotic Newcastle disease. Then coming to the neurotropic form. Neurotropic form is caused by the neurotropic strains. This disease is also known as the beach form, and it is characterized by the respiratory and nervous signs. Mortality is very high. Mesogenic form. Mesogenic form is also known as the Budet's form. It is slightly less virulent than the velogenic and neurotropic forms, but mortality may reach up to 50%. Egg production is dropped in this form. Then the lentogenic form. Lentogenic form is a mild form of the disease However, the presence of other pathogen, maybe viral or bacterial, may influence, and the birds may show symptoms similar to the velogenic strains. Asymptomatically enteric strain. In apparent intestinal infection, morbidity is usually high and up to 100%. Loss of appetite, restlessness, crop dilatation, decrease in egg production, etc. are also seen. Then then the symptoms are as seen in the pictures. The torticollis, it means the twisting of the head. Then the greenish diarrhea of the infected birds, depression of the birds. Then disease in the flock. In the flock, we will see the birds depressed, as shown in the picture. Then the lesions. The lesions are divided into gross lesions and microscopic lesions. The gross lesions include the hemorrhages in the esophagus or proventriculus, and proventriculus or gizzard junctions, and also in the duodenum, jejunum, ileum, but these are mostly seen in the virulent velogenic strains. Egg peritonitis, hemorrhagic or degenerated ovary may also be seen. Then the microscopic lesions include necrotic foci in the intestinal mucosa, lymphatic tissues, hyperemic changes in most organs including brain. Non-purulent encephalomyelitis may also be seen. This is the picture showing the hemorrhage in the proventriculus, inflammation in the lining of the windpipe, then hemorrhage in the cecal tonsil, hemorrhage in the intestines. Diagnosis as usual is based on the history and the clinical signs. Then the samples will be collected from live birds, include cloacal swabs, droppings, oronasal swabs. Samples from dead birds are brain, bile, liver, spleen, lung, kidney. But an important thing to remember at this point is that no lesion or clinical signs is specifically pathognomonic for this disease. So to confirm about this disease, we have to do isolation and characterization of the virus, which can be done only through serological tests, which include hemagglutinin inhibition test, ELISA test, RT-PCR, etc. The treatment. As it is a viral disease, Ranikhet disease has no specific treatment. The only vaccination, pre-vaccination, can ensure safety to the birds. However, for less virulent strains, antibiotics therapy may provide some relief to the birds. Prevention and control. Prevention is mainly by vaccination, and there are 3 types of vaccine available. There is live vaccine, example F. Lasota; live mesogenic vaccines, example Komarov, R2B, Mukteshar; and third is the inactivated vaccines. In addition, biosafety measures should also be followed, which includes proper sanitation, isolation, quarantine of the birds. Then the control at the national level should be done, like quarantine, slaughter, and vaccination of the birds which comes from foreign countries. And then at the farm level, strict hygiene practices and medical measures should be taken. So this is the vaccination schedule for birds, not only for Ranikhet but also for a number of diseases which are very important for poultry. Ranikhet disease, as you've seen, F1 or Lasota strain, and vaccine should be given in 2 to 6 days and repeated in 30th day. And R2B strain should be given in 6 to 8 weeks and repeated in 16 weeks. So the route can be given as intranasal, ocular, or subcut, as follows. So we've come to the end of this video. The references are taken from the textbook of Preventive Veterinary Medicine by Amalendra Chakravarti, and also from a textbook of Veterinary Special Pathology by J.L. Bhagat and A.K. Katiyar. Also, the references are made from the Tanuva's notes, and the pictures are taken from www.nadis.org.uk.com. So thank you. I hope this video will be helpful to everyone, and if you like this video, please share with your near and dear ones. Thank you.